Okay, kiddos, chapter 6, marine microbes, 51 slides. We'll stop at 26 rough, okay? All right, marine microbes. Key concepts. We'll let you look at those on your own time. So, marine viruses, quite appropriate that we should start with this. Significant source of food and diseases of eukaryotes. Marine viruses were first reported in the 1970s, and reliable counts were made in the 1980s. So if you've seen my little virus in the classroom, right, he had a purple head, hexagon shaped, and then with these little legs, right? And that's what this is showing right here. There's a hexagon shape, that stem, and then these little legs. The legs are so that they can latch onto the cell membrane of the host cell. The entire purpose of this capsule right here is just to hold its genetic material, be it DNA and RNA. It just really depends on which one of the two it's going to have, depending on the type of virus it is. Okay? Is a virus a living thing? That is one of the biggest debates in science right now. So let's review what are the characteristics of living things. They have to be, so C, D, E, G, there's 8 of them, H, M, R, R. They have to have cells, they have to have DNA, they have to evolve. So, by the way, viruses have cells, have DNA, evolve, they have to grow and develop, they have to have homeostasis, keeping a constant setup, be it pH, temperature, what have you. All right, salinity. All right, metabolism, that's it right there. That is what viruses don't have. They have to be able to reproduce. They do. What they actually do is the virus will go into the host cell, attach to the host cell, infect their DNA into the DNA of the host cell, and then the host cell will do its typical job, transcription, translation, copying its DNA, turning that DNA into proteins, and then all of a sudden, the host cell starts making a bunch of baby viruses. And that host cell busts open, releasing all those baby viruses who are now going to go infect other cells. And that's why you can wake up in the morning, feel perfectly fine, and come 4 o'clock in the afternoon, you're at death's door feeling horrible, because that one virus can come in and infect your cells and just take over your whole body very, very quickly. So they definitely reproduce. They need a host cell, but so do some of our parasites. So that's not what makes it non-living. And then respond to environment. It does respond to the environment. The fact that it does not have metabolism, the true metabolism, that is what is holding it up in the scientific community from being classified as a living thing. Rely entirely on hosts to reproduce themselves. Origin of viruses, there's 2 hypotheses: highly reduced prokaryotic parasite or renegade genes. We really don't know where they came from. One of the reasons they tell us not to feed manatees is because there's a lot of viruses they can contract from us. I don't know if you guys have heard, but COVID-19, we knew, obviously, it's been proven that it has come from the bats, from the eating of bats. I don't know if you'd heard it could get passed on to cats. It has now been proven that it can go from humans to dogs. And for the very first time, we have a case at the Bronx Zoo that a tiger at the Bronx Zoo has contracted COVID-19. He has tested positive for COVID-19. So that is the first tiger, first wild cat, large cat to test positive for COVID-19 in the world. So that was a sad discovery. I don't know what they're going to do with him. I don't know if they're going to treat him. Actually, I don't know if it's a boy or a girl. No. I don't know if they're going to put the animal down. I don't know. But it is a Bronx Zoo, so keep an eye on it. And so, anyways, to come back to this picture, that's why they're telling us be very careful with the feeding of animals, wild animals. Viral structure. You actually have different shapes of viruses. You have this simple capsule. You have a spiral. You have the one that you've seen in my classroom. It just depends. That's their life cycle we described. It comes in, attaches, infects, and then it will, what we call lyse. It'll burst the host cell open, thereby releasing a bunch of baby cells. It's very rapid. Infect, replicate, release. That's it. It's very simple. That's the lytic cycle. There is also the lysogenic cycle. It's the same as the lytic, but the viral nucleic acid may remain inserted in the host genome and reside there for a few cell divisions before it becomes lytic. So it'll sit in there and it'll stew and get stronger before it becomes lytic. Viruses are 10 times more abundant than any prokaryotes, and again, we've said that they are prokaryotes themselves are very abundant. Marine bacteria, general characteristics, they're prokaryotic. Remember, prokaryotes have no nucleus, no membrane-bound organelles. They have circular DNA. So, by the way, no membrane-bound organelles. That means they do not have no mitochondria, no chloroplast, no vacuoles, nothing that has a membrane, right? No nucleus, like we said. And their DNA is circular. They reproduce asexually by binary fission. Oh, look at this, beautiful. It's so pretty. I love these guys. Cyanobacteria, blue-green bacteria, they're photosynthetic. Not all bacteria are photosynthetic. These happen to be. They have chlorophyll A and B, carotenoids, phycobilins. They can be in single cells or in mats. Other photosynthetic bacteria can be anaerobic bacteria. Obligate anaerobes. Obligate means it must live without oxygen. Without oxygen. Okay? And then facultative anaerobes. That means they can live with or without oxygen. They can go either way. All right? Chemosynthetic bacteria. These guys are really cool. They are still going to produce glucose, but they do so without the presence of sunlight. They actually are going to take the sulfur, hydrogen sulfide right here, and they're going to be able to take that process and turn it into glucose on their own. It's really, really neat. Heterotrophic bacteria, decomposers, they undergo respiration and fermentation. When we say respiration, we mean cellular respiration. Let's go back real quick. So respiration, remember, is the opposite of photosynthesis. So it's going to be your C6H12O6 plus O2. And it's going to become water, CO2, and E, don't you dare be saying sun, because we don't have sunlight coming out of our butts, ATP, okay? These guys can help with consolidation, lithification, sedimentation. They can produce what's called marine snow, and it's the shower of organic material that's going to come down from the upper waters down to the deep ocean. And when we say the ocean current and the deep ocean comes down and it brings up things up along the continental shelf, right, and brings it up to the drop-off, right? There's a lot of fish at the top of the drop-off. That's because the currents are bringing that marine snow that fell down to the deep ocean back up. So it's that whole cycling. Nitrogen fixation process that converts molecular nitrogen that's dissolved in salt water into ammonium ion, which is a form of nitrogen that organisms can use, and then convert it into proteins, amino acids for the proteins, and into nucleic acids, a form to be used for nucleic acids. Only some cyanobacteria are capable of fixing nitrogen, not all of them. Endosymbiotic theory. I don't remember if we discussed this. Hopefully we have. Basically, the question is, how do we go from prokaryotes to eukaryotes? And the thought process is you had a large prokaryote that swallowed up a smaller prokaryote by endocytosis, right? It brought it in, and instead of consuming it, it actually just created a symbiotic, so endocytosis, and create a symbiotic relationship. And in doing so, this led to mitochondria. Well, what evidence do we have? Good question. Mitochondria has its own ribosomes. It has its own DNA, and it has two membranes. Now remember, every cell has to have four things, whether it's prokaryotic or eukaryotic. It has to have a cell membrane. It has to have its own DNA, ribosomes, and cytoplasm. Well, mitochondria has its own ribosomes, its own DNA, definitely cytoplasm, and it's got two membranes, which would be indicative of the first membrane it had originally, and then the second membrane that had been formed when it was taken in by endocytosis. The other organelle that has the exact same characteristics is chloroplast. So it's believed that chloroplast and mitochondria, both involved with energy conversion, right? Photosynthesis, cell respiration. These two were originally prokaryotic organisms that were absorbed by a larger prokaryotic organism, and instead of being consumed by the host, a symbiotic relationship was created between them. That's the entire endosymbiotic theory. Okay? You have bioluminescent bacteria, which have photophores, which is a light-producing organ in certain fish and other animals, found in the ink sac of squid. General characteristics of archaea, so archaebacteria. So remember, your prokaryotes are divided into two domains, bacteria and archaea. Let me try that again. Prokaryotes are divided into two domains, bacteria and archaea. Archaea, so the first ones were true bacteria. Okay, these guys are our extremophiles. They're usually very small. They're still prokaryotic. They are going to live in your extreme conditions. They're your extremophiles. Extreme salt, so halophiles. Extreme pH, acidophile, so typically acids. And then extreme temperature, thermophiles. Okay? Differences between from bacteria, cell walls lack special sugar amino acid compounds. Let me double check something really quickly. Discard. Ooh, sweet. We are right at one more slide, and we're right at the halfway point. And nutrition types. These guys can be photosynthetic, chemosynthetic, so that's your deep depths, so the ones that undergo chemosynthesis, and some can be heterotrophic. Most are methanogens. All right, and then we hit eukarya, and I'm going to stop here since we're at the halfway point so that the recording's not too big.